In the following, let's consider an amazing case of evolution of human placenta. This mystery was recently unveiled in the top scientific journal Nature. It's an amazing example of not Darwinian evolutionary event based on the gene flow, followed by a rapid adaptation via purely Darwinian process of natural selection. More details of this process are discussed in the following short video.
What if I told you that the DNA inside you isn't really your own, at least not all of it? Around 8% of all our genetic information is from viruses infecting us and leaving their genome inside ours. And the really weird thing is that you probably wouldn't be alive without one of these ancient viruses inside you. In fact, humanity likely wouldn't exist at all without an infection tens of millions of years ago. Several genes available in us. Our cells have made use of one viral gene in a really revolutionary way. The envelope glycoprotein coded by the ENV gene gives the retroviruses a way to fuse their outer membrane with the membrane of the cell they are infecting. The envelope glycoprotein, or ENV protein, recognizes a cellular membrane protein, anchors itself to the protein, and slips inside the cell. With the viral ENV gene adopted, cells gain the ability to fuse together. They can fuse together to make a sheet of cells that have multiple nucleuses and share all cell organelles. Though this only happens if the cells also have the membrane protein recognized by ENV on the cell surface. Both are needed for fusion. This sheet of cells opens up completely new possibilities for embryonic development, allowing for the formation of the placenta. More and more evidence supports that adopting the ENV gene turned our ancestors from egg-laying mammals to placental mammals, drastically changing the way the embryo develops. With the evolution of the placenta, an embryo does not need to be in an egg, facing the outside world, but stays safe inside the mother. The placenta is an organ connecting the developing fetus and the mother inside the womb. The fetus's blood flows through the umbilical cord to the placenta, where only a thin lining of tissue separates the fetus's blood from the mother's blood. Outermost on this very thin tissue layer is the syncytiothropoblast, a layer of cells fused together via syncytin 2 protein. Syncytin 2 is the name we have given to the domesticated ENV gene in our genome. The fused together syncytiothropoblast cell sheet is invasive and is what implants the embryo to the wall of the mother's womb. Later, the syncytiothropoblast facilitates the flow of nutrients and oxygen between the mother and the fetus. And its functions don't stop there. It also produces hormones, regulates the immune response, and protects the fetus from pathogens. We probably wouldn't be here if it wasn't for a rare event around 40 million years ago. A retrovirus infected our ancient ancestor, and its genome happened to end up in the germline cells and spread through the population. It inserted itself in chromosome 6, giving us syncytin 2. This here is what part of the human syncytin 2 protein looks like, using X-ray crystallography to determine its 3D shape. This here is not the full syncytin 2 protein. Its full structure has not been determined. This part here is key in the fusion of membranes. Syncytin 2 is not the only viral ENV gene that we have turned to our use. In a separate rare event around 25 million years ago, another retroviral infection gave us syncytin 1, another viral ENV gene that became part of us. While we have a model for how syncytin 2 fuses cells together to form and resupply the syncytiothropoblast with new cells, the role of syncytin 1 is not as clear. We humans have two virally derived syncytins, syncytin 1 and syncytin 2. Placental mammals are many, but very interestingly, most placental mammals don't share our synchetins. Instead, the incorporation of retroviral ENV gene for mammalian placental formation has happened over and over. At least 10 times are known so far. So far, a synchetin gene has been found in every mammal studied, with different synchetins fusing placental cells in different mammalian groups. A model proposed by Thierry Heidman and colleagues from the Gustave Roussy Institute explains the findings like this. The first synchetin gene domesticated came from a retrovirus infection around 150 million years ago. This synchetin gave rise to all placental mammals. The first synchetin has since been replaced by newer synchetins, unique to each mammalian group. Each new synchetin has given the species an evolutionary advantage. With enough time, millions of years, more rare germline infections leading to virus gene domestication have taken place, giving mammals a variety of synchetin genes. This model is supported by findings like the ENV-V synchetin, a domesticated ENV gene that seems to be crucial for placental formation in old world monkeys like macaques. The gene is found in the human placenta as well, but has lost its ability to fuse cells together. The human ENV-V could be a synchetin gene that has been replaced in humans by synchetin 1 and synchetin 2, but is still functioning in old world monkeys. Not only have retrovirus infections likely initiated and shaped the evolution of the placenta of mammals, they have done so in non-mammals as well. Recently, the Mabuya lizard, a lizard that has a placenta and gives live birth, was found to have a synchetin gene of its own. As with the synchetin genes of mammals, this synchetin was able to fuse cells grown in a cell culture and was expressed in the placenta. With the placenta evolving multiple times independently, it seems like a successful strategy for embryonic development.
Now, let's turn to our evolutionary reasoning. But in brief, let's look first at the Baltimore classification of viruses. The David Baltimore grouped viruses into families depending on their type of genome: DNA, RNA, single-stranded, double-stranded, and so on, and their methods of replication. Totally there are 7 families or 7 groups. Group 4 is notable for us because it comprises positive sense single-stranded viruses. So here is coronavirus, which is causing so much trouble to us right now. What we're talking about in this particular example is group 6 viruses, which are called single-stranded RNA retroviruses. And they have this RNA, which is transcribed into the DNA and inserted into the genome. And then from the genome, it's transcribed again into the mRNA from here to double-stranded DNA, and then from here to mRNA, which is used to translate the sequence into the protein. So this group 6 viruses, like HIV, for example, causing the AIDS disease, tend to insert their DNA into the into the host genome. And when this happens, of course, new genes are added. But if, let's say, the target cells are germline cells, then these changes can be propagated across the generations. The placenta as a trait was first emerged in the result of viral infection, which represents the gene flow event from virus to ancestral species of all amniotes and placentals. This is clearly not a Darwinian process, since the viral infection definitely not increased fitness of this affected ancestral lizard. It was lucky that it just survived amid rapidly collapsing population of its kin. This event drastically reduced population size and variation of traits in this proto-population. The genetic drift, which followed by pure chance, led to significant increase of protoplacental traits in this small population. As it turned out, in the longer perspective, this fusion of cells into continuous sheet was adaptive and was selected by subsequent Darwinian evolution in this population. Plausible evolutionary steps in Darwinian paradigm might be the following. So there are 6 main concepts, and I colored the statements according to the concepts presented earlier. So, number 1, species tend to overproduce, as always. So this leads to competition. Placenta enables to keep fetus inside the better temperature controlled environment, nutritional flow and oxygenation. It provides more flexible hormonal support of development and increases evolutionary plasticity of development. It means that more phenotypic variation might be generated by playing with the hormones. So the variation in the population came with retroviral infection of germline cell, perhaps in many individuals at the same time. The large functional gene was inserted into the genome. This is the source of variation. Not just a random mutation here and there in the DNA, but insertion of the entire new gene. So this addition of proto-synthetine gene helped formation of the placental membrane and perhaps also inhibited eggshell formation, so that the viable amnion stayed inside the body now. So this provided a huge advantage. This accelerated the development cycle, and as a result, the individual produced more offspring. Therefore, it increased its fitness in the population, because fitness in the population is the number of offsprings left. Since the gene insertion was in the germline cell, this new gene was passed down to the next generation. Natural selection provided positive selective pressure to increase frequency of this newly acquired allele in population. This resulted into 2 groups of individuals, which perhaps couldn't interbred anymore. This in turn resulted in speciation. So that is approximately how the reasoning looks like. In this lecture, in summary, we explored specific basic language and basic concepts of evolution using 4 recent humans events in evolution. We refreshed and discussed the 6 main concepts of Darwinian evolution. We saw how the 6 main concepts can be used to construct Darwinian evolution reasoning, explaining origin of a complex trait. So in the next lecture, we will explore evolutionary origin of humans and our place in the global tree of life. Thank you very much for your attention.